Hello, I'm Dr. Leslie Benson, a pediatric neuroimmunologist at Boston Children's Hospital. I'm really pleased to be here to present our work looking into autoimmunity in ROHHAD syndrome. My talk is titled Autoimmune Profiling Supports a Paraneoplastic Etiology in Pediatric ROHHAD. In the interest of transparency, I've listed sources of salary support and consulting that are completely unrelated to any of our ROHHAD work. As background, at times tumor may trigger an autoimmune process within the nervous system. These are referred to as a group as paraneoplastic neurologic syndromes. Basically, tumor tissue triggers and initiates a response from the immune system. That immune response may help to control the tumor, but also may cross-react at times with self-tissue, in this case, nervous system tissue, and cause injury. The red stars are indicating a common protein or target of that immune response that may cause injury to the self or normal tissue. The symptoms and consequences of this attack depend on which cells or tissues are targeted. Here is a model of of typical paraneoplastic neurologic syndrome with sort of the three pillars of what are required to call something one of these syndromes. The first is a a tumor outside the nervous system. Second is a classic clinical syndrome. And third is an antibody that is directed towards a protein within the tumor and the self tissue. Other features that are common to these syndromes include inflammation within the targeted tissue, inflammation within the spinal fluid, and a response to treatment. So immune therapy can help to calm things down and improve symptoms. One classic paraneoplastic autoimmune condition in children that is well established and accepted is called opsoclonus myoclonus ataxia syndrome, or OMS for short. This, like ROHHAD, affects young children and is associated with a neuroblastic tumor in about half of the patients. And in this syndrome, there is definitely a response to immune therapy, and treatment can improve outcomes. The piece that's missing from the paraneoplastic puzzle for OMS, however, is that to date, there's not been an identified antibody or target associated with the syndrome. So how does this relate to ROHHAD? Well, we have limited ROHHAD literature due to the rarity of the disease, but among the case reports and series that are out there, there are reports of inflammation within the target tissue, which here we think is most prominently the hypothalamus, but maybe other areas. There's definitely an association of tumors similar to OMS. There is reports of inflammation within the cerebrospinal fluid. There is certainly a very specific classic associated clinical syndrome. There are reports of a response to treatment, and I've listed some of the references here. What hasn't been found to date is a consistent antibody directed towards tissue in ROHHAD syndrome. So with that background, we have collaborated with labs at UCSF to look at the hypothesis that of an autoimmune etiology may underlie pediatric ROHHAD syndrome. And so working with Dr. Callie Mandelbrim, who led the basic science side of things, Dr. Mark Gorman and I provided the clinical information and samples, and together we looked at this question. And so using our cohort of 9 patients, 4 female, 5 male, all of whom had rapid onset obesity, all of whom would have met the criteria used previously in the 2007 Izzy Ludlow paper, and 8 of the 9 of them having a neuroblastic tumor. With these 9 patients, Symptoms started somewhere between 2 to 5 years of age, and they were diagnosed somewhere between 2 to 12 years of age. And the time to sample collection varied depending on when we were able to do so. So classically, method 1 is used to identify autoantibodies with paraneoplastic syndromes. However, all of these prior methodologies used rodent-based studies and did not work to identify an antibody in ROHHAD syndrome in our hands or others. So with the development of a new technology called FIP-seq, we were able to screen our patient's serum and CSF looking for a common antibody using a human-based programmable phage display platform. So what does that mean? I'm going to do my best as a clinician to explain this basic science. So what they do is they take the patient's sample, for in this case ROHHAD sample, expose it to this phage library that they've created where all these different viral phages produce their own little piece of protein, and all those proteins together add up to encode all the proteins in the human body, the whole proteome. And when those are put together, if there's antibodies that recognize self-protein in the patient sample, it binds to the phage who's expressing that protein that it's recognizing. And when they come together, they can be isolated out, and the protein part of the phage can be sequenced to identify which protein antigen bound to the human IgG. And so initially we piloted this with 3 patients, and were found a striking, uniquely enriched protein for this protein called ZSCAN1, standing for zinc finger and scan domain containing protein 1. It was identified in all 3 patients. And so we expanded our study to some additional samples on ROHHAD patients that we had available, as well as some controls, looking at normal controls, and obesity, neuroblastic tumor control, and OMS controls, many of whom, about half, had neuroblastic tumors. And we looked at serum and CSF where it was available. And what you're seeing here is that the amount that ZSCAN was very specific to our ROHHAD patients and was not seen in any of the other controls that we looked at here. And it also hasn't been seen in the other cohorts of patients that are both published and unpublished that have been tested through the Derisi lab. And we're talking about other paraneoplastic diseases, other non-neurologic autoimmune syndromes, as well as patients with cancer. So with that finding, they went forward and validated the finding in cell-based assays, as well as radioligand binding assays, and again reproduced that ZSCAN1 antibodies were present in 7 of our 9 ROHHAD patients. So what is ZSCAN1? Well, what we know is it's an intracellular protein. We don't know what the specific function is, but we do know that it's primate specific and does not have a similar protein in rodents. According to protein atlases, it is expressed in the brain and most highly in the hypothalamus, but also in some endocrine tissues, and interestingly, the pituitary gland. We were able to obtain healthy human hypothalamus from donation, and validated that ZSCAN1 protein is present in human hypothalamus. And we used one of our ROHHAD neuroblastoma samples to confirm that ZSCAN1 is also expressed in ROHHAD neuroblastoma, ROHHAD-associated neuroblastoma. So in summary, we have an autoantibody to an intracellular protein called ZSCAN1 that are present in 7 of 9 patients with classic ROHHAD clinical phenotype. We have ZSCAN, so here we have ZSCAN1 expressed in the target tissue, healthy human hypothalamus, as well as ZSCAN1 expressed in the tumor specifically. So in conclusion, Anti-ZSCAN1 antibodies are a promising biomarker for ROHHAD syndrome. They are the first defined intracellular antigen associated with a childhood paraneoplastic neurologic disorder. And what's big for our field as well is that human-specific approaches can identify novel antibody-associated neurologic syndromes that prior methods couldn't. And as always, for the ROHHAD research is definitely in need. I've included here a link to the preprint article for those who are interested, and I have to acknowledge our funders as well as my many collaborators. So as I mentioned, Dr. Callie Mandelbrum led the basic science along with collaborators there in the Derisi Wilson and Pleasure labs, and my mentor Mark Gorman contributed to the conceptualization and results from this study. I also want to thank ROHHAD Fight and the Shore Foundation, who provided specific targeted funding for these projects. Thank you so much for your time and attention.